Welcome to another episode of Tom Kennedy Science. I'm your host, Dr. Tom Kennedy. Now today we're going to talk about mutations. You know, those things that created the X-Men. I'm just kidding. However, you people that can drink milk and are lactose tolerant, you're a mutant. But I wish I had your mutant powers to drink milk because I cannot. And that means I can't eat ice cream. Now what exactly is a mutation? So mutation is a change in your DNA, right? I mean, anytime you change the sequence of your DNA, you have a mutation. And they were discovered a long time ago, over 100 years ago, by a scientist named Thomas Hunt Morgan studying fruit flies. And he discovered mutations. So he's out one day and he's like, that fruit fly has a white eye. What's up with that? And then from there, they were able to figure out that mutations are the ultimate source of variation. And that's important, right? Because we need mutations. I mean, we always think that mutations are bad. But the reality is mutations, they create variation in a population. And that is what natural selection acts upon, right? I mean, you think about like Game of Thrones, right? I love Game of Thrones. It's not exactly the most kid-friendly movie in the world. And I definitely have issues with some of the things they pulled. But that being aside, there are some really cool things that they did. The Baratheons, they had black hair. You know, the Lannisters had their golden hair. And the Targaryens had their white hair. And these traits, all these differences in the traits, are caused by mutation. So when we look at humans and we see people with black hair, people with black skin, or white skin, or different colored skin, or different colored eyes, or different blood types, that variation is caused by mutations, right? So mutations have led to all the variation we see amongst humans today. Sexual reproduction is taking a deck of cards and reshuffling it in every generation. But you're just playing with the same deck over and over and over again. You know, some card hands are good, like, an ace, king, queen, jack, and a 10 all suited. You're going to win. You got a winning hand. You're Einstein over here. If you're playing with a 2-7 split, you're Beavis and Butthead. You might as well fold them, right? Okay, but you want a new card in the deck. That is where mutations come into play. And mutations, they create new alleles. So like Mendel's pea flowers, the white flower is a mutation. O blood is a result of a mutant form of A blood. Mutations can be small. They could be as small as I'm switching out one nucleotide for another. I'm losing a nucleotide. To mutations can be, entire genomes are getting duplicated. And as a result, mutations also run the gamut of completely silent, completely neutral, have zero effect on the organism, and they could never have an effect on the organism, to good, to creating a new allele that might be useful, to slightly bad, or good or bad, depending on the environment, to so catastrophically bad that the organism dies within a few rounds of cell division after the zygote is formed. So let's talk about these. Let's talk about some of these different types of mutations. And I want to start off with like the smallest ones first. Let's talk about a single nucleotide substitution. A single nucleotide substitution can have all kinds of an effect, or none. If you remember, the genetic code has got some redundancy built into it, right? If you take, for instance, the amino acid alanine, as long as you have G, C, guanine and cytosine, it does not matter what the third position is. It could be thymine, adenine, guanine, or cytosine. Doesn't matter. The genetic code is redundant, especially for alanine. So any mutation where you do a single nucleotide substitution, I switch out a T for an A. If it's in that third position, that third codon for alanine, it's a silent mutation. It has no effect on the organism because you still make alanine. So we would call that a silent mutation. So a lot of mutations, just straight up, not going to have any effect, especially in that third position. Now, you start switching around in the first or the second position of a codon. Remember, a codon has 3 nucleotides that code for an amino acid. The first 2 are the most important for determining the amino acid because the third position is a little bit redundant there. But if you start messing around with the second or third codon, you're going to get an amino acid that's different. So let's take a look at sickle cell disease. Sickle cell disease is caused when you have improper folding of a protein, and it causes the blood cells to become sickle-shaped, especially in people that are homozygous for this. Sickle cell disease is caused by a single nucleotide substitution. That's it. One single nucleotide substitution. Basically, what happens is a T, a thymine, is switched out for an adenine, and that's in the second position of the codon. And instead of getting the amino acid glutamine, you get valine. Okay? That's it. That is how a single nucleotide substitution can cause a major change. There are other ways too. Remember, there's only 61 codons, right? So we've got 61 codons coding for 20 amino acids. But there are 64 of these possible combinations of nucleotides. It turns out that 3 of them are stop codons. So if you do a nucleotide substitution and you get what is called a premature stop codon, so you get, I'm going to use RNA here, UGA. I always remember that. I think University of Georgia in Athens, Georgia, right? UGA is a stop codon. Let's say I've got UGC, and I switch that C out, that cytosine out for an adenine, and I get UGA. I now have a stop codon. And it doesn't matter if you're 5 amino acids into making your protein or 1,000 amino acids. Translation stops. Now, translation occurs at the ribosome. That's when we take the information that's stored in our RNA, and that's translated into a sequence of amino acids. And if you come across one of these premature stop codons, it stops translation, stops making that protein immediately. So it truncates it. So you may or may not get a functioning protein depending on how much of it was made. So for example, a premature stop codon, you have O blood. O blood is actually a mutated version of A blood. So you start translating, you start making that surface antigen for the A, but about a third of the way in, you reach a stop codon and it stops and you don't make the antigen. So O blood basically has a truncated A, and your body doesn't recognize it as an antigen. So we always learn Mendel's pea plant experiment, and we learn that he's got these white flowers and these purple flowers. The color purple is caused by this pigment called anthocyanin. Now, DNA doesn't code for a protein called anthocyanin. Anthocyanin is a pigment that requires proteins to make it. However, a single nucleotide substitution causes a mutation where the cell doesn't make anthocyanin, and as a result of a single nucleotide substitution, you create a new allele. In this case, the recessive version is white. So it's recessive because you're not making anthocyanin, so if you get 2 copies of it, you don't have any anthocyanin, you don't have any of that blue pigment. If you get only 1 functioning copy, well, you make enough anthocyanin that your flowers look purple. Now we're starting to see the molecular connection from the patterns we see in heredity to what's actually happening at the level of the genes and at the proteins. So there you have it. There's a great example of how you can get these different alleles caused by a single nucleotide substitution. Some of them you get them from switching out one amino acid for another, and another one is you could have a premature stop codon, which basically knocks out that protein. All right. Well, there are other simple types of mutations. Those are insertions and deletions. Now, insertions and deletions, depending on the size of them and where they're formed, can have anywhere from no consequence to big consequences. Other ways that insertions or deletions works is they change the reading frame. Okay, now I'm not very good at doing this here off the cuff, but imagine I've got the word Gattaca. That's a great movie with Ethan Hawke, Jude Law, and Uma Thurman about genetic manipulation in the future. It's a very good movie from the 1990s. I know it's a little bit dated for a lot of you because it's older than you guys are, but it's a very good movie. We're actually facing the issues of genetic engineering now. But let's say I get that word Gattaca. That carries information. Now, if I do a deletion and I remove the letter G, I now have Attica. Not only do I have Attica, but my entire frame shift occurs. Or if I do an insertion where I've got T Gattaca, remember codons are like in groups of 3, what happens is downstream of that insertion or that deletion, every single amino acid changes. So in this case, insertions and deletions, they can be absolutely catastrophic for that protein. Now, if it's a protein like a pigment, Hey, a white flower also attracts pollinators. No big deal. If it's a protein that knocks out something like cytochrome oxidase inside of your mitochondria, then you can no longer use cellular respiration. The embryo will never survive past a few rounds of a few rounds of mitosis before it dies. Okay, so we've got single nucleotide substitutions, insertions, and deletions. Those are important because, like I said, they often make new alleles. And they can be good, they can be bad, they can be neutral. There are other types of mutations that are also important. One of those is gene duplication. Now, a lot of people have criticized the evolution of mutations, going, how in the world do we get a world teeming with complex life with these random mutations? And they're thinking of these random A's changing to a T's or a T changing to a C. What they don't realize is that we actually have what's called a gene duplication. Now, there's this process of creating our gametes where we have meiosis, right? Meiosis has 2 rounds of cell division, blah, blah, blah. But in meiosis, all the chromosomes come together, right? You got the sister chromatids from your mom, your sister chromatids from your dad. They come together and they form the tetrad. And it's in that tetrad formation that you actually have proteins involved with swapping out alleles from your mom's chromosome and putting them on your dad. Now, sometimes these chromosomes are not totally lined up. They're a little off. And when they're off or something goes not quite right, you can have a gene duplication. So let's say a good example here is you guys have dogs, right? Or you've been around a dog. Dogs live their life through their nose. I mean, they smell everything, right? Well, it turns out that dogs have an incredibly good sense of smell. And to have a good sense of smell, you need proteins in your brain called your olfactories to receive these different odors in the environment, these different chemicals. The more protein receptors you have, the more different chemicals you can have. Well, mammals, the ancestors, have over 1,000 of these genes. And they are a gene family caused by multiple rounds of gene duplication. So every time you duplicate a gene, it's subject to mutations. And then once it's subject to mutations, it creates these different alleles. And if they're bad, their natural selection removes them from the population rather quickly. But if they're helpful, they're kept. And so like the mammalian sense of smell is really good, especially in dogs and most other mammals except primates and cetaceans, which are whales and dolphins. I'll come back to that. But that sense of smell is so good because of a gene duplication of that gene, creating a large gene family, allowing us to detect many odors in our environment. Now, here's another example of this. Our eyesight. We have pretty good vision compared to most other mammals. We see a lot more colors than they do. And the reason why is because early in primate evolution, there was a gene duplication in the proteins of your eye that detect light, and they of course mutated. And guess what colors we can see? Reds, right? We can see reds and oranges and all these different colors that a lot of dogs don't see very well. It's not that dogs have bad vision. Some do. But it's not that they have bad vision. They just can't see the number of colors we do because we have a more complex visual system. And we don't have the most complex visual system. That goes to the mantis shrimp. They have like 16 genes to see color. I can only imagine how they see the world. But let's get back to this gene duplication that occurs during meiosis. This creating multiple genes at once, you don't have to start from scratch. This has been incredibly important for allowing complex life to evolve. And in fact, we have these things called Hox genes. They're the developmental and regulatory genes. We'll talk more about them later in a different episode. But they're like these master control switches, and they regulate all these other genes. And in mammals and invertebrates in general, we see these gene duplications of these master control genes that allow for additional complexity. So we don't rely on simple insertions, deletions, substitutions for building up complexity. It's these gene duplications. And then also, if you have gene duplications, guess what? You can have full-on chromosomal duplications and full-on genome duplications. That's rare. It doesn't always work. It worked in frogs and in a few plants, and that once again allows for a lot more genetic material for life to work with. Now, once you get these whole genome duplications and and chromosome duplications, there's this cool thing in evolution where they kind of whittle back down into fewer genes. So that, in a brief explanation, is mutations. Like I said, we're not creating the X-Men here, but we are creating variation in a population. We are creating gene families that allow for the evolution of complexity. And understanding the sources of mutations is also important for understanding things like cancer and stuff like that, and how we can treat that. Because if we can understand what's causing the cancer, then we can prevent exposure to those things, like world's worst #1 cause of cancer is smoking. Yeah, it causes all kinds of mutations inside of your lungs. Okay, well, that's been another episode of Tom Kennedy Science. Until next time,